Dankeschön.
Все. Пожалуйста. Это последняя лекция. Все.
Thank you. Well, dear students, today we will be telling about reactions of protein synthesis. Before it we talked about proteolysis and metabolism of amino acids. Today about synthesis, storage and transfer of genetic information. And today's lecture will be dedicated so-called central dogma of biology, transfer of information in living systems, structure and function of nucleic acids necessary to repeat it, organization of genetic materials in cells, replication of DNA, and a little about methods of investigations of nucleic acids. And all of it dedicate appearance of proteins, synthesis of proteins. Really, proteins is main functional molecules in living organisms. Without proteins, life impossible in form as we understand it. Proteins, main functional molecules, but information about it stored in DNA, nucleic acids. It's take place opinion that any billions years ago was RNA world, when RNA was its storage and realization of information, realization of functions. But now proteins more effective functional molecules. And in our body we have near 150,000 of different proteins. It's so-called proteome, and information about it coded in genome. But the number of genes near up to 22,000, 21,500 genes, so-called genome. You can to see number of genes much less than number of proteins, and it different reasons lead to it. But one gene usually not every time have information about more than one polypeptide structure. And the proteome is a very changeable set of proteins, because it's very thin, very flexible balance between proteolytic digestion and protein synthesis. Now you can remind that lifetime of proteins in the body, proteins in the region from any seconds to some months of different proteins. And now you can see a picture of different genes, functions of products of different genes. You can see multiple functions of proteins. It may be enzymes, big but not abundant group. It may be different regulative enzymes, enzymes and proteins for function of regulation and activity for nucleic acids, and other transformation of proteins, structural proteins, and more. Ion channels, immunoglobulins, you can see transporters. But you can see very big part, some less than half, but also very big part of proteins, function of it unknown. Genes and correspondent products, function unknown. Now it percent gradually decrease. It not maybe more than 40%, but really it big part proteins and genes with unknown functions. But it important, essential for life, because in genome it take place present. And genome in was discovered and determined fully in 2003 year. It results of two teams. One team is so-called Human Genome Project, which will be combine of near 20 laboratories from different countries. You can to see list of laboratories with main participation in determination of full genome. And it was second team. It was private team of Craig Venter. It's very original and talented scientist. And now we know that human genome consists near 3.1 billion nucleotides. It's haploid, but because it's self-present diploid set of genome, it 6.2 billion nucleotides. And is it more or not? For haploid genome, you can to see mammals consist genome in region more than from 2, 3 to 9 billions nucleotides. It's not most abundant. Sometimes protists, for example, you can to see plants, amphibians have much more sometimes, more than 100 billions nucleotides. But mammals genomes, including humans, much more multiple, much more different variants of genes present. Most complex, maybe human genome, most complex. And transfer of information in living organisms, for all living organisms in which live in our planet, described by so-called central dogma. Central dogma of biology, based on storage of DNA, or storage of information in DNA, RNA, or intermediate carrier, or maybe sometimes end product, for example, ribosomal or transfer RNAs, and proteins. Proteins main functional units. And the central dogma was named it result of, it was called by Francis Crick, very famous, effective, brilliant scientist, really. And it 3.1 billion nucleotides distributed in pieces, 8 pieces called chromosomes. Chromosomes, we have 22 pairs of autosomes and 1 pair of sex chromosomes, XY, XX, and XY, XY for male. You know it very well. And genetic information distributed in each chromosome uneven, no, it's not randomly, but it distribute according by evolutionary roots. So now you can to see set of chromosomes with indication of expression sites. Different place on chromosomes, more or less important for storage of genetic information, and expression of each gene have different intensity. For example, we can to see there it density in the content of genes in different chromosomes. Biggest size of chromosomes first and second, and in first chromosome you can to see a lot of more than up to 450 genes. minimal size of chromosomes, you can to see 21, 22, but number of genes relatively high. But sometimes at 18th chromosome, number of genes relatively low, up to 30. In X chromosome, sex chromosome, you can to see a lot of number of genes. In Y chromosome of male, number of genes up to 50, and it transfer in generation male line. It can be checked by history of evolution of males and checking of distribution and changing of genes in X chromosome. It indicate about evolution of females. But average density you can to see 12 genes on 1 million nucleotides. Yes, well. And a little about history. About history, at first describing of possibility of and quantitative determination of transfer of information, phenotype, it was described by Gregor Mendel. And you can see 1965 it was described. It discreteness of inheritance properties. And the first association with presence of any chromosomes was described by Thomas Morgan. He was not effective biologist, but it was also effective so-called cytologist, morphologist, which can to describe presence of chromosomes. But nature of chromosomes is also unknown, and first describing of nucleic acids was described by Friedrich Mischer in 1969, but it has described, but it was unknown function. And before beginning of 20th century, it was idea that nucleic acids, monotone repeating monomers like for example, glycogen and polysaccharides, any other polysaccharides, without any specific functions. But function of DNA was described by Frederick Griffith with when he can to transfer of properties of microbial colonies, microbes, Rauth, so-called in pneumococcus, non-toxic, non-infection, to smooth, so-called S variant with S infections, and Rauth non-infection. And using of and transfer of nucleic acids can lead to transfer of properties. And it was evidence of it was supported by investigations by Oswald Avery, Colin MacLeod, and Maclean McCarthy by Nobel Prize. This was in the 40th years of last century. And later, nature of and details of structure was described by multiple investigations. For example, very important, Erwin Chergaff with described correspondence between purine and pyrimidine nitrogen bases in DNA. And Rosalind Franklin and Maurice Wilkins described that DNA, winded structure, and James Watson and Francis Crick describe structure of open described structure of DNA, double strand, and possibility of transfer of its information and coding, mechanism of coding of its information. And now it's described by so-called central dogma of biology, when DNA storage form, transcription it synthesis of RNA, and translation it synthesis of proteins. But now it added to it reverse transcription from RNA to DNA and replication of DNA in viruses, RNA viruses. And no, we talked about it. Distribution of genetic information associated with special places, special pieces of DNA, which call gene. And DNA, now you can see known function of DNA to only storage of information, but RNA more multiple. It's storage and transfer of biological oxidation. Sorry, storage of biological information. Yes. Well, and what about structure? Structure of DNA and RNA is sequence of nucleotides. And now you can see there each nucleotide consists a combination of sugar, ribose or deoxyribose, nitrogen bases, and phosphoric acid. It's stable structure, relatively stable. Sometimes maybe addition phosphoric acids. It's mono phosphate, diphosphate, 3-phosphates, and D and 3-phosphates combination of negatively charged groups lead to increasing of free energy of its substances. And nucleotide 3-phosphates is source of energy, effective source of energy. But it also components of polynucleotides chains. And about nitrogen bases. Nitrogen bases, again necessary to repeat, it derivates of pyrimidine or purine bases. Pyrimidine base it heterocycle, one cycle, six atoms nucleotides with 2 nitrogens. Pyrimidine and oxidized parts of 2 oxygen bound with pyrimidine, deoxypyrimidine will be uracil. Methylated uracil is thymine, and aminated 1 oxygen instead of 1 oxygen amino group, it will be cytosine. Cytosine and thymine in DNA, cytosine and uracil in RNA, cytosine only cytosine in both in DNA and RNA. And the purine bases. Purine base it double cycle. It pyrimidine plus imidazole cycle. You can see totally 4 nitrogens, and combination nitrogen carbon nitrogen carbon nitrogen carbon nitrogen, you can see, is very effective functionally and for stability and for functional activity. And we have 2 derivatives of purine, it adenine, aminopurine, and guanine, amino and oxypurine. And possible 2 variants of distribution of electron density, so-called lactam, when carbon bound with oxygen, and oxygen only with carbons, and NH, nitrogen bound with hydrogen, or lactim, maybe lactim and double lactim form, when carbon bound with oxygen and oxygen bound with hydrogen. Really, it's a resonance structure with constant exchanging between lactam and lactim forms. But lactam form much more effective, consists much less free energy, and in any 100 times more preferential presence in lactam form. But if will be transformed to lactim form, it will lead to impossibility of doubling complementary association between classic combination thymine adenine and guanine cytosine. Double lactin form will lead to destroying of it variants. But lactam more stable and lactam more frequent variant of interactions. And classic interactions correspond to lactam form. It's little detail, but it's source of usual native natural reasons of mutations, by the way, one of it. More than classic nitrogen bases possible, a lot of in DNA and RNA can be present so-called minor nitrogen bases. Usually it methylated forms. So, for example, methylcytidine, methyladenosine, methylguanosine, and sometimes methyl group will be oxidized, hydroxymethylcytidine. And maybe dominated form inosine, pseudouridine, methylguanosine, thiouridine. Sometimes present, rarely, but sometimes it pseudouridine, for example, present in transfer RNAs. Yes. And the methylated form of nitrogen bases, it can be signals for regulation of gene expression in DNA. Now with combination of combination of groups in nucleotides, it can be RNA when presence of ribose, and DNA with deoxyribonucleotides with missing of OH oxygen in second carbon. And of course, DNA will be more hydrophobic, less hydrophilic because absent one polar group. And additionally, in DNA present methyl group, also hydrophobic. In RNA, it's absent. It's also additional factor for more hydrophility for RNAs. How it will be appeared, these nitrogen bases? It's complex reactions which will be beginning from ribose 5-phosphate. And ribose 5-phosphate, if you will be remind carbohydrate metabolism, it's one of products of pentosophosphate pathway. And ribose 5-phosphate, it's precursor of all nitrogen bases. Not only nitrogen bases, nucleotides. And synthesis nucleotides lead to from ribose 5-phosphate, and at first phosphorylation. Again, necessary must be high energy substance. And you can see that phosphoribosyl pyrophosphate, precursor of O, appearance of pyrophosphate group associated with first carbon. And later, sequence of reactions when glutamine, 2 molecules of glutamine at first, and the glutamine next, again, donor of amino glycine, no, glutamine, yes, present. Glutamine, glycine, derivative of glycine, 1 carbons. If you will remind metabolism of glycine, a lot of substances can be derivatives of glycine, and the glycine is a donor of single carbons important for anabolic reactions. Now you can to see glutamine, donor of amino group, glycine bound there. Again, formic residue, also derivative of glycine, with participation of derivative folic acid, vitamin BC, tetrahydrofolic acid. Later, glycine next, again formulation by again derivative of glycine, imidazole ring, later aspartate like cycle of urea cycle, urea synthesis, and again as a result will be appearance of fumarate, very similar with urea synthesis. Next, again formulation and cycling in ring. Inosine monophosphate with oxygen instead of amino group. Changing to amino group will be lead to adenine, or association with amino group on this carbon will be lead to guanine. It's synthesis of purine bases. You see, and ribose and phosphate will be present. These complex reactions lead to purine bases. For pyrimidine bases, also necessary to have phosphoribosyl pyrophosphate. This is one. But synthesis of pyrimidine ring appeared from interaction of carbon dioxide and amino group, now from glutamine, and synthesis of carbonyl phosphate. Well-known carbonyl phosphate, you met it in urea synthesis, but it takes place in different space of cells. Urea synthesis beginning in matrix of mitochondria. Now it carbonyl phosphate by carbonyl phosphate synthase 2 will be appeared in cytoplasm. And next, again binding with aspartate, very similar again with the synthesis of urea cycle, but in different space and in different, with participation of different enzymes, and forming of so-called orotic acid. You can to see it pyrimidine ring plus carboxy group, pyrimidine with 2 oxygens, and missing of carbon dioxide lead to uracil, UMP, uridyl monophosphate. Next, it may be amination to lead to cytosine, or methylation lead to thymine. And sequence of nucleotides in DNA and RNA, you can to see it interaction by phosphate groups between ribose or deoxyribose of different nucleotides. It will be phosphate, for example, ribose, phosphate, and more. And but it polar distribution. At last, it linear structure, and at last, on one side of this strand, we will have free phosphate group non-associated with next ribose or deoxyribose, and on other side will be free OH group non-associated with the next phosphoric acids. And now you can to see now it's chromosome, it's chromosome 14th. Length more than 100 million nucleotides, but it's linear structure, and it will be 100, for example, 50 million nucleotides, but at last we will be achieved 5' end on first end, 5' end. And last, later, moved on phosphate, ribose, deoxyribose. And at last will be achieved 3' end with absence of phosphate group. It polarity of sequence non-equal, and reading every time beginning from 5' end and finishing by 3' end. You can to see that. And about DNA. DNA double strand. Then 2 strands orientate one to other by and associated by hydrogen bonds, complementary interactions, classic interaction. A interact with adenine with thymine, and guanine with cytosine. Complementary sequence. And you can to see sequence alternative, direction of sequences alternative. One strand on one side consists 5' end and finished 3' end on another side. Alternative complement sequence orientate alternatively 5' end where was first 3' end and 3' end finished where was 5' end, anti-parallel. And again, very important, lactam form. If will be transformed to lactine form, interaction will be imperfect, but more preferential, and will be repeat, lactam more preferential. And double strand of DNA stabilized by at first hydrogen bonds. Increasing of temperature will be lead to destroying of hydrogen bonds, and additionally it so-called stacking interactions by hydrophobic interactions, because mainly it structure hydrophobic, and stacking interaction additional factor. And next additional factor of stabilization, it negative charged groups of phosphate which will be orientate outside of nucleotides. And DNA it rarely acid with a lot of phosphate groups orientated and localized on the outer surface of DNA. And not only simple double strand, but in DNA can be present so-called palindromic sequences when sequence of nucleotides alternative of one strand, and it lead to forming of loops and stems in place of palindromes. And it's important, one of reasons of signals for DNA. Sometimes it may be very complex interactions. Now you can to see combination of stem loops in RNA with importance of its configuration for functions, for transfer of information or forming of correspondent secondary and tertiary structure of RNA. Now you can to see microbial 16S ribosomal RNA, multiple stems, multiple loops, and all of it lead to very unique configuration which will be recognized by enzymes which work with RNAs and participate in transfer of information. Now by the way, about types of RNAs. No, for students it's well known messenger RNA. It carries a carrier of information for protein synthesis, but also transfer RNA. It's a carrier of amino acids for protein synthesis. Ribosomal RNA, structural function for forming of ribosome and function of providing of its functions, but also a minimal set added so-called small nuclear RNAs. Its complex with certain proteins lead to importance of excision of introns from messenger RNA in our genes. You know, a lot of sequence senseless sequence without functional now in this moment without functional activity importance, and the excision of it provided by including small nuclear RNA and the so-called microRNAs. It short double-stranded RNAs with the possibility of regulation of gene expression, because microRNAs can recognize of messenger RNA, and it will be signal for specific nucleolytic enzymes, nucleases in cytoplasm. In our cytoplasm, a lot of nucleases, but it will be inactive before interaction with microRNA. And microRNA, it will be labeled for target messenger RNA, and nucleases will be digest it mRNA with limitation of translation. It minimal set of messenger of RNAs in cells, total RNAs in cells, and it number of it gradually increase and increase. And now about relative sites of DNA and other intracellular parts. For example, you can see hemoglobin and DNA, thickness of DNA near 2.5, 2.4 nanometers. It's compatible with cell membrane thickness, compatible with usually not too high level, not too high molecular weight proteins. Sometimes it's usually more, but hemoglobin, example, and water, much less amino acids and other. You can see relatively big structure, not very long. And DNA, it very it's chemically active substance, and because it constantly present in environment with chemically active environment, possible random spontaneous modifications of its amino acid radicals. Nucleotide, nitrogen-based radicals. Now you can see cytosine free oxidation of oxygen, oxidative deamination. You are a big specialist of deamination, for example, and changing of amino group to oxygen lead to transformation of cytosine to uracil. And if it was methylated, methyl cytosine will be transformed to thymine. Thymine is normal, usual normal nitrogen base, and it also a natural reason for mutations. Guanine oxidation will lead to xanthine, and adenine to hypoxanthine. Very important and possible also depurinization. Purine bases binding of an glycosidic bond with ribose with association with purine bases relatively slow. It can be easy broken hydrolyzed by water, intracellular water, and it leads to depurinization. And in each cell can be any hundreds, sometimes any thousands of purine bases can be desquamated, can be digested, spontaneously hydrolyzed, and it also must be reparated by corresponding enzymes. Next, modifications. It alkylation by alkylation agents. For example, smokes, tobacco smokes. Modification lead to spontaneous, non-planned, for example, methylation. It lead to changing of properties, of course, and changing of possibility of creation of complementary sequence. Next modification, very serious cancerogenic substances, it's so-called benzpirene. Benzpirene, it's substance which present in any, for example, including tobacco smoke. Active substance which can be easily modified So now with purine-based guanine. And covalent binding lead to this dysfunction, impossibility of transfer of information, creation of complementary sequence. Next modified agent is aflatoxin. Aflatoxin is the result of synthesis in, for example, decay products of apples, for example. Decay apples, it's dangerous source of aflatoxin. Really, aflatoxin is not toxic, but toxic is derivative. And derivative of aflatoxin created by microsomal oxidation, cytochrome P450, can lead to different variants. If it will be creation epoxy hydrase, it lead to non-toxic substance. And maybe if it will be conjugate with glutathione, it will be lead to forming of non-toxic substance. But sometimes it can be lead to modification of nitrogen bases. And in this case, it will be lead to destroyed storage and transfer of information from this place. Next mutational agent, it ultraviolet. Ultraviolet is non-pleasure because it absorbed by pyrimidine bases, and when if two pyrimidine bases will be localized one to other, it's possible forming of covalent binding between pyrimidine bases, and it will be lead to, of course, destroying of transcription of its genetic information and replication. Now you can to see result of interaction between two thymines. And reparation, all changing of mutations lead to activation of system of reparation. Reparation possible in any cases. It can be mutation in mutation of nitrogen base. In this case, its part will be excised by glycosylases, and maybe more short or more long place will be digested, and filling of empty place, in this case, by DNA polymerase, and restoring of it. If it will be absent of reparation, in this case, in period of replication of DNA doubling, system complex of enzymes will be stopped in this place, but in this case also possible complex will be dissociated, and components of complex will be lead to reparation of its place and reactivation of next replication of this. But accumulation of mutations will be lead to destroying of normal replication and expression of genes, of course. And most dangerous for function of cells, it's so-called double-strand breaks. And reparation depends on cycle of cell. If in cells will be present complementary homologic chromosomes, it will be lead to possibility of rare synthesis, reparation of full sequence of nucleotides by so-called homologic recombination. When two homologic chromosomes will be lead to exchange forming of this synthesis combination of two chromosomes, so-called forming of so-called holiday junction, and there is reapparation of missed sequence. But if double-strand chromosome, it take place, homologic recombination take place in phases S or G2. But if cell present in G1 stage of cell cycle, when homological chromosomes is absent. In this case, it will be realized so-called non-homologic end joining. With the missing of it, double strands will be associated, but it will be lead to missing of any part of genetic information. Again, it really natural reason of mutations, with missing of any part of genetic information. And mutations and dysfunction of enzymes of reparation lead to serious destroying of cell functions and very often reasons of oncotransformation. Now you can to see mutated or inactivation of any enzymes lead to development of different variants of cancers, and where and how it will be observed and what symptom complex you can to see there. And of course, all of it will be realized across oncotransformation of tissues. No more detail will be studied on Department of Oncology. Well, and next part of today's lecture is packing, organization and packing of genetic material in cells. For it, we will be tell after break. Now, please break near 7 minutes, please. Well, так, dear students, next part of lecture is about packing. You can to see if we will be stretched out of DNA, length will be up to 2 meters. But how it will be packed in structure in nucleus, which we cannot to see by eyes. It result of combination with correspondent proteins and forming of chromosomes. And proteins which can interact with DNA, it so-called histones. Histones very interesting proteins, conservative. We have a lot of copies of histones, and and uh, we have 5 main types: H1, H2 histones. Yes, H1, H2, A, H2B, H3, and H4. And H2A, H2B, and H3 and 4 forming lead to forming of octamer, 2 subunits of each, with and DNA winded on each octamer, up to 2 winds on each octamer. Altogether will be called nucleosomes. And now you can to see there, and linker path of DNA between nucleosomes, and each part can be interact with the histone H1. I hope you know it, but now all of it repeat. And interaction with interaction of DNA with histones, forming of nucleosomes, important for possibility of an expression of genome, corresponding genes. Now, what about histones? A lot of mass near equal that DNA in nucleus, and how it can to interact with DNA. DNA is acid, and if will be separate DNA from to water by dissociation of proteins, it will be stack, like, because the same charged groups of phosphates will be lead to take out one to other, and creation of very long stack. But for folding of it, the same charged, negatively charged groups necessary, neutralizate its negative charge. For it necessary positive charged amino acids. And it lysine, arginine, histidine, you can to see. And it consists a lot of it positively charged amino acid radicals, which lead to neutralization of negative charge phosphates group of DNA. And it will conservative sequence. And we have some copies of genes, and we have sometimes maybe different variants between this set of primary structure of histones. And histones not only structural elements, but it also sensitive elements for checking of DNA structure. Mutations, any mutations will lead to destroying of interactions between histones and DNA. Now you can to see nucleosomal strand, by the way, which also folded in next order of winding. It electron microphoto, and noted about you can to see a little information about structure, number of amino acid radicals, molecular weight, and number of lysine and arginines. We talked about it. And again, well-known picture when folding of DNA in chromosome, it any orders minimum 4 4 orders of folding: double strand, nucleosomal, betal string, chromatin strand, compactization of chromatin strands, and next order of folding, minimum 4, sometimes maybe more, forming of metaphase chromosome. We can see only in metaphase plate. But really in living organism, in living cells, a lot of genetic materials decompactizated, and to lead to possibility of interaction of DNA with correspondent factors of transcription. Next important modifications of histones, modification of acetylation of tails. C end, carboxy end of histones orientate extra nucleosomal space. And acetylation and deacetylation, modification of amino acid radicals, will be lead to a more compact or less compact packing of nucleosomal strand. Now you can see there non-acetylated, unacetylated. It's compact. Acetylated forms lead to less compact interactions, and possibility it will lead to possible interactions of enzymes with DNA. And now you see place of regulation. Now with promoter region. Promoter region is a place where will be beginning transcriptions of genes. It must be not covered by nucleosomes, localized between nucleosomes. Now, for example, transcription factors will be interact with DNA, and in this place will be open for enzymes. Histones will be dissociated, but when its factors will be dissociated, it depends on environment, and it depends on state of DNA and other. And in this case, it will be possibility of transcription will be absent. Now, for example, factors of transcription factors. How this very long DNA must be must be synthesis of complementary sequence, doubling in period of S phase of cell cycle. How it can be provided? It's called replication, and in microbes with cyclic DNA, it takes place on one place, only site. But for eukaryotes with very long, very big DNA, it beginning from multiple place, some hundreds place of origin of replication, and it will be moved one to other. Forks of replication, and it leads to relatively short period of doubling, in contrast with one site of origin of transcription of replication in microbes. But microbes consist short, much less nucleotides in DNA, genetic material. You can see it replicational bubbles, replication fork, which will be moved one to other, and special enzymes at first at helicase, which will be lead to unwinding of DNA, and DNA polymerase, which will be synthesize new DNA. And elongation and synthesis of DNA possible only on 3' end. Elongation and synthesis take place from 5' end to 3' end, which will be oriented alternatively to template DNA. And one strand will be synthesized constantly in this case, but second will be synthesized by pieces. And constantly synthesized will be called leading strand, by pieces will be called lagging strands. And continuous synthesis and discontinuous synthesis. And again, synthesize every time from 5 to 3 prime end. Why? Because for synthesis of any polymer, necessary source of energy. And the source of energy will be the same source of mononucleotides. And for every time, for each addition of next one nucleotide, necessary corresponding nucleotide 3-phosphate. And cleavage of pyrophosphate lead to addition of new nucleotide to elongated chain. Now you can see there. For elongation, necessary nucleotide 3-phosphate, for example, for binding of adenine, deoxyadenine, necessary deoxynucleotide 3-phosphate, for deoxynucleotide deoxyadenine, deoxy ATP, deoxy TTP, thymine 3-phosphate, deoxy CTP, cytosine 3-phosphate, and deoxy GTP, guanosine 3-phosphate. For synthesis of 3 billions nucleotides, necessary 3 billions equivalents of ATP. And the source of energy every time from 5 prime end. Source of energy for elongation on 5 prime end is absent. And it leads to polarity of movement and polarity of synthesis, synthesis of corresponding elongated chain. Next, very important, if it will be elongated, take place on one direction, ahead of upstream of synthesis will be a period, can be a period supercoiled DNA. It will be lead to destroying of synthesis. And for avoiding of it, previously, before synthesis, necessary to provide unwinding of DNA. It provided by special enzymes which call total name, it topoisomerases. It can be helicases and gerases. Topoisomerases, total name because topology is a region of mathematics which will be described properties of surface. And it two types can be divided. First and second type. All of it nucleases, but one type when will be digested and reassociation by rotation of two strands simultaneously. It's first type. Second type, it when one strand will be digested and around its polynucleotide chain will be transfer across constantly present chain by only digestion on one strand, and will be decrease number of windings. Now, all together lead to possibility of unwinding of double strand of DNA. Helicase will be lead to digestion, but when will be appeared one strand DNA can try to reassociation backside of gerase, and also one strand DNA is very sensitive to hydrolysis. Can be spontaneous hydrolysis of phosphoester bonds. For avoiding of it, backside of helicase will be binding of special low molecular weight proteins, which call single-strand binding proteins, which will be prevent of reassociation. And next, possible synthesis of DNA, but DNA polymerase, it enzymes without possibility of binding with one-strand DNA. It very serious enzymes with possibility, enzymes of reparation, really. It can synthesize complementary sequence when present one polynucleotide strand will be present, but cannot bound, recognize and bound with one-stranded. But RNA polymerase can bound with one-strand DNA. And next enzyme, it will be RNA polymerase, which will be synthesized short part of RNA, which called primer. And when will be appeared double-strand DNA, in this case, DNA polymerase will be displaced RNA polymerase and bound, and ahead of it, in template DNA will be appeared empty place, and DNA polymerase will be provide synthesis of complementary sequence of DNA. Again, you can to see elongation of 3' end. One will be directed to fork, a second will be on other side alternatively. It will be appeared next empty place on one strand. Next, fork go ahead, and one strand will be elongate constantly, but second will be empty, and next binding of RNA polymerase, next binding of DNA polymerase, and elongation. And you can to see constantly synthesize. A leading strand will be synthesized constantly, but lagging strand will be elongated by synthesized by pieces, and these pieces called Okazaki fragments. But the DNA polymerase 1, which main enzyme which will be synthesized, provide bulk of synthesis, cannot exist primers, RNA primers. It result of function of next enzyme DNA polymerase, which will be digest of RNA primers and fill it these empty places. And connection last phosphate pair bonds will be provided by DNA ligase. Yes, and result. You can to see it list of participants. It minimal list of participants necessary to have DNA template, DNA helicase, single-stranded binding proteins, which will be prevent reassociation, primase, it RNA polymerase, which can be synthesized short primer of RNA, DNA polymerase 3, which will be main enzyme for synthesis, DNA polymerase 1, which will be digest primers, and DNA ligase, it will be enzyme which will be provide interaction between Okazaki fragments. And all of it moves with velocity near tens, some hundreds nucleotides in prokaryotes and tens nucleotides in eukaryotes. Now we can to see DNA polymerase, it dimeric form, which will be surrounded of DNA and move it along it. And DNA polymerase move it in one direction, and for lagging strand, it will be any correspondent loop which constantly changing length. It can be described more or less a long chain, and it can be described by trombone model, trombone-like model. Yes. Now we can to see, and the single-strand binding proteins constantly associated with the leading strands. Now you can to see rate of elongation, some hundreds for prokaryotes and some tens for eukaryotes, because in eukaryotes present histones. About method of investigations. For investigations of DNA, now it's possible for each human can to determine own DNA. It's not too high price, up to 1,000, for example, dollars. For it necessary to have DNA, extraction of DNA, and after this, different variants, maybe multiplication of DNA. At last, it was cloning in prokaryotic or yeast organisms. Or now it effectively provided by using of PCR, polymerase chain reaction, and determination of details of structure by electrophoresis, different variants of electrophoresis, or Gill electrophoresis, or maybe capillary electrophoresis, or maybe direct determination of sequence by new generation sequence methods. No, at first, most ancient view method, it was development of DNA with restriction enzymes, and multiple fragments were separated by electrophoresis, and separated picture was transferred on membrane, nitrocellulose or acetate cellulose membrane or capron membrane, different variants. Paper towels used for increasing soaking of solution, which consists DNA fragments, to membrane. And later development of blood. Blood better to use because it leads to fixation of separation picture. Development with DNA probes, labeled DNA probes. It may be radioactive at last, but now it used probes labeled by fluorescent stains. And now we can to see method of separation in agarose Gill. No source of power supply, Gill, agarose Gill, and chamber of electrophoretic chamber. And now it's stained samples moved, and later it determined by irradiation by ultraviolet, for example. And the polymerase chain reaction, very effective method for separation, for determination of presence of corresponding genes in corresponding genome. For it, previously, you must know what you want to determine. And after determination of target, separation of target, necessary to have two set of primers, which will be flanked, localized at the ends of target part of genome. One will be oriented 5' end. This is template 3' end on this side. Primary oriented 5' end, and synthesis possible on this direction. Second, on this direction. 3' end on this side, primer on this direction, and move it today. And synthesis of primers provided by typical serious method of organic chemistry. phosphoramidite method, for example. Its length must be near 20 nucleotides. If less than 20, it will be decreased specificity. Possible this variant of sequence in this genome may be multiple. More than 20, usually more than 25, sometimes it may be up to 30. It will be lead to some problems with the interactions. Will be decreased sensitivity maybe. Well, and after this, when you have primers, when you have template DNA, necessary to have set of nucleotide 3-phosphates, source of synthesis, and polymerase, DNA polymerase. After separation of strands one to other, primers will be bound with corresponding target place, and DNA polymerase will be part of double strands and will be elongate on direction 5' end to 3' end. And after any time will be a period of place for synthesis of primer by correspondent primers with correspondent chain, correspondent strain. Yes. First will be go ahead, far from there, but will be a period place for synthesis of second primer. And on next steps will be a period result of synthesis with correspondent previously separated length. And after second multiplication stages, again dissociation, binding of primers and elongation. Dissociation, binding of primers and elongation. And on each step will be doubling of synthesis with correspondent with the same molecular weight. Now you can see now high temperature. Next, decreasing temperature, primers bound. Next, synthesis of complementary sequence. Again, separation of strands, binding of primers, synthesis of complementary sequence, and more and more. And as a result will be appearance of multiple. You can calculate number. It will be billions oligonucleotides with the same properties. And later necessary to determine the presence of it by electrophoresis. But it unknown what present in inner space between primers. It can be very important. In this case, it may be development with specific enzymes which will be specifically recognize and digest of its sequence, or maybe using of fluorescent label, fluorescent probe, which can be indicate bound with multiplicated part of genome and synthesis of fluorescent probes. And the determination of primary structure can be provided by sequence of nucleotides by a method of Friedrich Sanger. For it used D-deoxynucleotide. Deoxynucleotide absence of OH group on second, but now it absence of OH group in third also nucleotide absent on second and third carbons in ribose. In this case, when nucleotide will be appeared in space of synthesis, it will be stop synthesis because next phosphoric acid cannot bound with OH group of deoxyribose. And the method based on separation of system reactional volume on 4 test tubes on one test tube present all necessary for synthesis, everywhere present all necessary for synthesis. But additionally, in first will be added a little dideoxy dideoxythymine, dideoxy ATP, dideoxycytosine CTP, dideoxy GTP, and dideoxy TDP thymine. In this case, will be take place synthesis everywhere, but randomly will be appeared next dideoxynucleotide and will be stopped. And in this test tube will be appeared set of nucleotides, oligonucleotides, with stop synthesis on correspondent nucleotides, and set of nucleotides with different lengths. And now you can to see possible to directly reading of sequence adenine, guanine, thymine, cytosine, guanine, adenine, guanine, cytosine, and more. Now you can to see Frederick Sanger with this list of labeled zones with possibility of direct reading of sequence. Some thousands possible to determine by synthesis. But now it used by fluorescent labels with using of capillary electrophoresis. But idea is the same. Separation by size, and will be correspondent sequence can be reading. And it methods now useful for determination. This different weights. Or maybe so-called pyrosequencing. Pyrosequence is based on pyrophosphate, which will be digested from nucleotide 3-phosphate, will be hydrolyzed by pyrophosphatase. And if it will be used for registration of flash of light by enzyme, which will be transformate energy to pyrophosphate energy to light, to flash, we can to read sequence by registration of flash in different places of two-dimensional plate. Or maybe it provided by microarrays. Now you can to see this list of cells with correspondent probes, labeled probes with sequence complementary different parts of nucleotides, and development of mixture of DNA to this plate. We can to see fluorescent and non-fluorescent regions. It will be correspond different sequence of nucleotides, and this variant lead to possibility of registration of expression different groups of genes. For example, for determination of malignant cells. Next generation sequence, it more effective determination when DNA will be divided on short pieces of with length near 100 nucleotides, will be ensured to zones, to probes, which will be ensured to glass or non-soluble plates. And now you can to see this is a lot of primers which will be bound with non-soluble plate, and elongation with registration of flash of light, different nucleotide 3-phosphates labeled with different colors of flash. And we can to see plate with constantly changing of flash. And it possible direct determination of sequences and multiple sequences with length near 100 nucleotides. But problem is next, necessary to reconstructed of total sequence by registration of combination with the same for different variants of oligonucleotides. And in this case will be appear some problems, because if you have a limited set of points, you can to get with high possibility known shape and view. But if you have a lot of dots, realization of a picture depends on sometimes fantasy of investigator. And next generation of determination, it after separation of DNA pass across whole with size compatible with diameter of DNA, of valence strand DNA, and the registration of electroconductivity across this hole. And each nucleotide, when we will pass across this hole, will be individual influence on electroconductivity. And it also leads to possibility of direct registration of polypeptide chain. Now you're going to see there. And it very convenient method, but it lead to next problem for storage and defense of personal genetic data. If you can easy to get own genome, it's not too high price, but now in a situation of transfer of information between humans, it high possibility of leaking of its information, and it's real problem now. Thank you very much for your attention. On next lecture, we will be tell about transcription and translation. Goodbye.